Bismillahirrahmanirrahim. Today I am going to discuss about translation process. This is the figure from Lippincott. This page, next page is also from Lippincott. I hope you will study from Lippincott textbook this translation process. Now I am going to discuss in details elaborately the translation process. Now, at first, visualization problem. Just need to wait. Okay. Here you follow. This nucleotide base is mRNA. This is the mRNA 5 prime end to 3 prime end. This mRNA we get from transcription process, and this is tRNA, and this one is small ribosomal subunit. This green color is small ribosomal subunit. This is tRNA, and you also follow that this one is a large ribosomal subunit. As we discussed in discuss prokaryotic translation process, that's why it is 60s ribosomal subunit and it is 40s ribosomal subunit. So ultimately total 80s ribosomal subunit, not 100. 60s and 40s total 80s ribosomal subunit, not 100. Remember it. Now, at first, this small ribosomal subunit combined with this mRNA 5 prime to 3 prime end, and starts scanning of the initiating codon. This initiating codon, when is scanned or found by this small ribosomal subunit at the 5 prime to 3 prime end of the mRNA, when AUG is detected by the small ribosomal subunit, then its anticodon come with this tRNA. It means tRNA contains this anticodon. AUG anticodon is UAC. AUG is the initiating codon that is detected by or scanned by this small ribosomal subunit, and its anticodon containing tRNA is this one. This tRNA contains this anticodon and come to this site. And we also know initiating codon AUG anticodon that UAC. This tRNA contain methionine. So when teacher will ask you AUG for AUG which amino acid is specific, you answer that methionine. For AUG, methionine amino acid is specific. So methionine containing tRNA come to this site, and its anticodon or its codon is UAC. So AUG anticodon is UAC containing tRNA come to this site with methionine. So ultimately you follow initiating factor. Initiating factor is small ribosomal subunit with methionine containing tRNA is ultimately from pre-initiating complex or pre-initiation complex, and this pre-initiation complex bind with this mRNA. I discussed step by step how mRNA small ribosomal subunit starts this translation process and how pre-initiation complex is formed. Pre-initiation complex actually formed by tRNA small ribosomal subunit and GTP. Here GTP is consumed. GTP and initiating factor is formed ultimately from this pre-initiation complex. And at first, small ribosomal subunit scan this initiating codon. This initiating codon has anticodon UAC. This UAC containing codon tRNA is tRNA come to this site which contains methionine also. So ultimately here pre-initiation complex is formed, and the next step, in the next step, when codon with anticodon containing tRNA is fixed, mRNA fixed, then large ribosomal subunit come to this site. It means this formation occur and large ribosomal subunit come to this site, and ultimately formation of initiation complex. So pre-initiation complex, then initiation complex or initiating complex. So now tRNA with tRNA with mRNA, large ribosomal subunit, small ribosomal subunit, initiating factor ultimately form the initiation complex. So first step was pre-initiation complex. Now in second step is initiation complex is formed, and you follow large ribosomal subunit. Here you follow this one is large ribosomal subunit, P site, A site, and E site. P site, here you follow when large ribosomal subunit come to this site, P site is occupied by the tRNA. This one is P site. A site is empty, E site also empty. Now next step. Next step is just that this ribosomal subunit or this initiation complex move forward, but mRNA is fixed. mRNA does not move. Remember it that mRNA does not move, but initiation complex as well as pre-initiation complex move forward, but mRNA does not move. You remember this. Now you follow the next step, third one. uh, Here follow that at the previous picture there was a site was empty. You just follow here that in this figure just show that methionine, here was methionine, and here next tRNA come to this site with phenylalanine tRNA come to this site. At the next step phenylalanine come to this site. This one is the next step, third step. Here you follow third step. So this phenylalanine tRNA come to this site and combine with this initiation complex. So next tRNA, first tRNA with methionine come to this P site, but next tRNA with phenylalanine or other specific amino acid enters through the A site of the ribosomal subunit, and this process continues. So suppose another amino acid come to this site, try to come to the A site, and methionine must go to this E site. This phenylalanine containing tRNA must go to this A site, P site, and new amino acid containing tRNA come to this A site. So new amino acid containing or new tRNA come to enters through the A site, and tRNA from the A site go to the P site, and tRNA from the P site go to the E site, and tRNA from the E site come out from this initiation complex or from this ribosomal subunit. It means this tRNA come out from this initiation complex and recycling occur. It is just the next step. This occurring step by step, that amino acid with tRNA come to this A site or enters through the A site. tRNA from the A site go to the P site to the E site, and go out from the initiation complex. Here you follow that this amino acid come from this P site, now to the E site, and E site to the outside of the ribosomal subunit or complex. When tRNA go out from this complex,